How do fast-twitch fibers compensate for the lack of mitochondria compared to slow-twitch fibers? And could the difference between the two explain why we fatigue faster at higher intensities of exercise? In this video, we will look at a recent study on the mitochondria inside of slow-twitch and fast-twitch fibers, how they work, and how they seemingly evolved to collaborate and take advantage of each other's strengths. So let's dive in. So the paper we're looking at here is called Need for Speed, Human Fast-Twitch Mitochondria Favor Power Over Efficiency, a study from January of 2024, and I've put the link in the description if you want to go and read it in more detail. So let's start exploring. First of all, we can see that human skeletal muscle differentiates into two major fiber types based on the myosin heavy chain content, each with highly specialized contractile and metabolic profiles. So essentially we have the slow twitch fibers on one side and the fast twitch fibers on the other side. And it seems like slow twitch fibers are optimized for efficiency from an energetic standpoint. They have a lot of mitochondria. We'll see this just after. And they have less contractile elements, the myofibrils. Whereas the fast-twitch fibers have more contractile elements and less mitochondria. And we can see at the bottom here, they talk about the distinction between type 2A fibers in animal models and in human models. It looks like in animal models, type 2As are typically grouped together with the type 1 fibers, so they're part of the slow twitch family. But in humans, because of their twitch speed, the type 2A fibers are typically associated with the type 2X fibers, so they're put in the fast twitch category. So let's move forward. We're going to look at those two types of muscle fibers, slow twitch muscle fibers, which are typically red. Why? Because they contain more myoglobin. Myoglobin is the molecule that transports oxygen, stores and transports oxygen within the muscle cell. It's kind of the small cousin of hemoglobin. If you look at a hemoglobin molecule and you break it in four, you pretty much get a myoglobin molecule. And in fast twitch fibers, we also call them white fibers because they have very little of this myoglobin and obviously less oxidative capacity as well. Now, we talked about the twitch speed earlier, and what's interesting, even between the fast and the slow twitch fibers, is that they technically produce the same amount of force, but they do so in different time frames. So the slow twitch fibers take more time to contract compared to the fast twitch fibers, and that also means that the power that is produced by the fast twitch fibers is much, much higher compared to the slow twitch fibers. And to go back to the previous slide where we discussed the contents, relative contents of different fast twitch and slow twitch fibers, we can see here on this diagram that we have more capillaries around the slow twitch fibers, less capillaries around the fast twitch fibers, more mitochondrial volume inside the slow twitch fibers, about threefold difference that's pulled from that same study here, a threefold higher mitochondrial volume in slow twitch fibers. But on the other side, we have a lot more contractile elements inside the fast twitch fibers. So those are the main differences. Obviously, the enzymes that go with the different energetic pathways are also different. We have more aerobic, quote unquote, enzymes in the slow twitch fibers, more glycolytic enzymes in the fast twitch fibers. And those are the differences here. Then we can think about the electron transport system, which exists inside the mitochondria, actually on the inner mitochondrial membrane. And this is where the NADH and FADH2 from the Krebs cycle are going to be utilized. And the electrons will be transported from complex 1, the yellow one on the left, to complex 2, 3, and then 4. And oxygen is actually going to intervene at complex 4 and form water from what is going on here. And then what's happening in parallel to those electrons being transported throughout this chain is that the protons are being pumped from the matrix to the intermembrane space. This is what creates the electrochemical gradient, and then the protons are being pushed back down through ATP synthase, complex 5, with the green one on the right-hand side. And this is how we put ATP back together inside the mitochondria. And what it seems like from this paper is that the fast twitch and the slow twitch fibers don't actually have the same proportion of the different complexes that we find on this electron transport system. And if we go into a bit more detail, it says up here that the higher fat oxidation or fatty acid oxidation and oxidative phosphorylation capacity in fast twitch fibers is primarily due to an elevated capability to metabolize succinate. And this is done at the complex 2 of the mitochondrial or the electron transport system. As for protein expression, the 2 dehydrogenases of the electron transport chain, NADH dehydrogenase, which is complex 1, and succinate dehydrogenase, which is complex 2, were differentially expressed in the 2 fiber types when related both to our mitocontrol. So what we see on the graphs here on the right-hand side, the first one is that there is more complex 1 expression in the slow twitch fibers and more complex 2 expression in the fast twitch fibers. And if we put that down with an image, we could see at the bottom here that the slow twitch mitochondria have more of the complex 1 and the fast twitch mitochondria have more of the complex 2. So that's one of the main differences that we find between the mitochondria and the slow twitch fibers and the fast twitch fibers. And then we can go a step farther and look at what's happening with complex 5, the ATP synthase, and it seems like fast twitch mitochondria express double the ATP synthase levels of mitochondria in slow twitch fibers. As you can see here, the complex 5 at the bottom in bright red, there's more of them in the fast twitch mitochondria, and you can see the shape of the cristae is also different, right? So we have different folding of all those proteins and of those membranes inside the fast twitch mitochondria compared to the slow twitch mitochondria. And it seems like that's one of the ways that fast twitch fibers optimize for the lack of mitochondria. So they have less volume, but they compensate by expressing different proteins and changing the way that they function, if we want to keep it simple. Then there's another interesting quote here. It says that our results of increased complex 2 respiration in fast twitch fibers may explain the increased O2 cost per produced watt during heavy aerobic exercise, causing a nonlinear relationship in oxygen consumption during the final minutes of gradient exercise test to exhaustion. And this is probably linked to the VO2 slow component, although it does seem like the VO2 slow component is linked to some extent to inorganic phosphate accumulation, and this kind of drives ATP consumption up. And I can come back to this in another video if you're interested, but it's probably also related to what they're citing here, the fact that the increased complex 2 expression and respiration explains the increased O2 cost. So if we look at how oxygen consumption evolves at different intensities, and here we have the 4 intensity domains: low, medium, high, and very high intensities. We have stable VO2 at low intensity, and right from the heavy or the heavy domain or the medium intensity that we see here in orange, instead of reaching a steady state, there's kind of that little bounce up, and this is what we call the VO2 slow component. So this is probably what they're talking about here when it comes to those fast twitch fibers, and we'll see why just after. And essentially, if you progress that even further and you go above threshold 2, you cannot stabilize that slow component anymore, and this is why you tend towards VO2 max or VO2 peak. And then in the extreme domain, you reach task failure before reaching VO2 max. And so the way we can conceptualize that is by thinking about the Henneman principle and how motor units are recruited over the intensity spectrum. Essentially, you have the smaller motor units on the left, the larger motor units on the right. The smaller motor units have a lower activation threshold, means they need less neural input or neural output to be recruited, whereas the larger motor units have a higher activation threshold and they only get recruited later or farther up on the intensity spectrum. And this is usually the case that small motor units that have the low activation threshold on the left-hand side are attached to slow twitch fibers, and the large motor units on the right are attached to fast twitch fibers. We're going to keep it that simple, and there's some cases where it doesn't work exactly like this, but to understand how different fiber types are recruited across the spectrum, it's a useful model to use. And we can conceptualize it in that way as well. Type 1 fibers get recruited from the get, and then starting at medium intensity, we get some type 2As that are being recruited. And again, that seems to also contribute to that VO2 slow component that we saw on the other slide, and then the type 2X fibers that are recruited towards the higher ends of the severe domain and the extreme domain. And you also see differences in how the VO2 slow component is expressed depending on somebody's muscle typology. So if you have more fast twitch fibers, it looks like you have a bigger slow component. And if you have more slow twitch fibers, you have a kind of a smaller slow component. So I do think that those things seem to make sense when it comes to what the paper says and what we understand about that oxygen consumption kinetics across the intensity spectrum. Then we can keep going. A metabolic state that's really interesting may be occurring at the onset of anaerobic respiration, where the fast and slow twitch fibers exchange lactate for succinate to favor their respective electron transport chain composition. The succinate from slow twitch fibers could then act as a substrate for fast twitch fibers tailored towards succinate oxidation. Thus, fast and slow twitch fibers might exchange lactate and succinate during heavy exercise to symbiotically favor their respective predisposition towards complex 1 or complex 2 respiration. So, said simply, they collaborate. The fast twitch and the slow twitch fibers collaborate, and they essentially say, hey, you're better at using that stuff. Here, let me give it to you. And then, oh, you have some of that stuff that I'm better at using, so give it to me and I'll use it. So this is really interesting how those two fiber types work together. And we know that during exercise, the fast twitch fibers, if we talk about lactate, for example, they're net lactate producers, and slow twitch fibers are net lactate recyclers, if we want to call them that. So essentially that explains that transition between those that substrate of lactate, and then conversely with the succinate as well, which will be available in the type 1 fibers, but that get passed back to the fast twitch fibers, and they each have their role and how they work. And if we come to the end for the conclusion here, in conclusion, human fast and slow twitch muscle fibers host distinct mitochondrial populations. Slow twitch mitochondria favor sheer mitochondrial volume. We saw about 3 times more, and rely on complex 1 for efficient oxidative coupling and NADH oxidation. By contrast, fast twitch mitochondria are tailored for speed rather than efficiency through faster succinate metabolism and significantly elevated complex 2 expression. The fast twitch mitochondrial proteome may enable faster and more efficient mitochondrial morphology changes during high energy demands, likely as a compensatory mechanism for low total volume. So essentially, the fast twitch fibers have less volume of mitochondria, but they seem to essentially breathe faster or breathe more per unit of mitochondria. And this is also what we see when it comes to different training adaptations. And we typically say that sprint interval training, which is extreme domain exercise at the very, very high of the very, very top of the intensity spectrum, we see that it tends to increase mitochondrial respiration. So essentially, how much oxygen is being consumed per unit of mitochondria is improved by sprint interval training interventions. And that would be explained because during that very, very intense sprint interval training, we're going to recruit and fatigue those fast twitch fibers, which are kind of oxygen consumption oriented from a respiratory rate standpoint. But at the other end of the spectrum, if you think about the adaptations that you see from low intensity training, zone 2, continuous training, we see obviously adaptations in the slow twitch fibers, and we see an increase in mitochondrial volume density. So all this kind of holds together and jives with the other information that we already have and understand regarding different fiber types, where they're recruited on the intensity spectrum, and how they adapt to different exercise interventions. And we can also look at muscle fiber efficiency and how it varies between the fast twitch and the slow twitch, with the slow twitch being very hard to fatigue because, again, they're more efficient, but then the fast twitch being very fatigable, and they're unable to maintain their initial contractile force when they're repeatedly stimulated over and over again. So here's for this study review with a few extra slides thrown in. If you like this kind of content, please let me know in the comments. I'd be happy to make more videos of this kind. I've got lots of papers in the bank to look at with you. Let me know what topics you'd like me to cover. And if there's anything I said in the video that you don't agree with, I'd be happy to chat with you in the comments. Thanks again for watching, and I'll see you in the next one.